Next one that are found in the plasma is acute phase reactant or acute phase proteins, APP or APR, both are right. These are basically a group of proteins whose plasma concentration will change in response to inflammatory reactions such as infection, surgery, trauma, myocardial infarction, malignancies, and so on. This includes C-reactive protein, alpha-1 acid glycoprotein, fibrinogen, haptoglobin, alpha-antitrypsin, albumin, and transferrin. It is very important for you to know which protein's level will increase in acute phase and which protein will not increase or decrease in acute phase protein. So this chart is very important and high yield. Please do remember. In acute phase reaction, the proteins such as C-reactive protein will increase mainly. See here, 1000-fold amount. Alpha-1 antitrypsin will increase, alpha-1 anti-chymotrypsin will increase, ceruloplasmil, fibrinogen, haptoglobin, these all will increase. Some proteins will decrease in level as well, such as albumin, transferrin, retinol binding protein, these will basically decrease. So please do remember which proteins will increase during acute phase reaction and which proteins will decrease. So first one is the C-reactive protein. This is a major content of the acute phase protein. So sometimes in order to evaluate any acute phase response, we usually measure the amount of CRP. As we have seen, CRPs will increase in a very high amount in cases of inflammation, acute inflammation, okay, and will ultimately reach to the peak around 48 hours. The normal level of CRP or C-reactive protein is less than 0.8 milligram per deciliter. Functions of the C-reactive protein is initiation of the process of opsonization and phagocytosis. So they play roles in defense mechanism basically. They will activate complement, activate neutrophils and monocyte as well as macrophage. So mainly they will be able to recognize any microbial organism and will initiate the host defense system, the C-reactive protein. The next one in acute phase protein comes is fibrinogen, also known as clotting factor 1. Their role is to mainly take part in blood coagulation, and they also act as a precursor for fibrin, a substance that is required for clotting. They are usually activated by thrombin. Usually fibrinogen will be converted into fibrin by cleavage of the arginine-glycine peptide bond. They are also synthesized in the liver. Their normal value is around 200 to 400 milligram per deciliter. Now, this is all we had for our plasma proteins. So before we begin with the next content, let me first tell you what are the important things you should be knowing in the plasma proteins. Okay, first of all, what are the total proteins? The definition, very important, you should be knowing. You should be knowing all types of plasma proteins that we have, such as albumin, transerythrin, retinol binding protein, and so on. You should be knowing their features, their functions, and the clinical significance we have. Most important content, you should know the definition of acute phase reactant, in which proteins will increase during acute phase reactions and which will decrease. Okay, so those were the high yield contents of the last content that we just finished, known as plasma protein. Now the next content that we will continue is porphyrins and hemoglobin. Okay, in order to understand porphyrins, first we need to know what is pyrrole. So let's see what are basically these pyrrole. So pyrrole is an aromatic compound which are made up of 5 rings basically, and they have the formula of C4H4NH. You can see the structure over here. They will have 5 rings, which closed rings. It's an aromatic compound. Porphyrins are formed by these pyrroles. To be more precise, they are formed by joining of 4 pyrrole rings with the help of methynyl bridges, and will have a metal ion linked to the nitrogen atoms of the pyrrole rings. The most important, biologically important porphyrins are existing as a conjugated proteins. They are present usually as a conjugated proteins, and these basically includes hemoglobin, myoglobin, cytochromes, catalase, peroxidase, and tryptophan pyrrolase. So basically, porphyrin will be formed by 4 pyrrole rings which are joined together with the help of methynyl bridges. Okay, now let's see some details about this thing. First one, hemoglobin. It's a type of porphyrin there, which is in a conjugated protein, which is defined as a conjugated protein and made up of 4 subunits. Each of these subunits have a heme group and a polypeptide chain. The heme group of the hemoglobin is basically made up of the porphyrin and the ferrocide. Hemoglobin has the ability to bind with oxygen reversibly and therefore allows them to transport oxygen in the body. Myoglobin have a similar structure just like hemoglobin, only difference is that they are monomer. Myoglobins are found in muscles and they also can reversibly bind with oxygen. Cytochromes. These also contains iron porphyrin which are conjugated with the protein. And their iron porphyrin portion is quite similar to that of the hemoglobin, and they are found basically in the respiratory chain. Also we call it the electron transport chain in the mitochondria. Some of these cytochromes may also perform other functions. For example, microsomal hydroxylation. The fourth one is known as an enzyme. It's called catalase. This also has iron porphyrin and mainly they are found in animals. Their role is to detoxify hydrogen peroxidase. Fifth one we have is called peroxidase. Mainly this is also another enzyme and their role is also to act on hydrogen peroxidase, and they will mainly be found in plants basically. Number 6 are tryptophan pyrrolase, again an iron porphyrin containing enzyme. They will act on the amino acid tryptophan. Now, this is how a typical pyrrole will look like. Okay, first of all, pyrrole we know is something like this, which will be made up of how many? 5 rings with an NH group here. Their formula was C4H4NH, if you remember. Okay, so 4 of such pyrrole rings will together join to form a porphyrin. And this is how the structure of porphyrin basically looks like. Okay, the parent porphyrin is also known as porphyrin. Okay, you can see here we have 1 pyrrole ring here, 1 pyrrole ring here, 1 pyrrole ring here, 1 over here, 1, 2, 3, 4. They are joined with one another by methynyl bridges. So there is a scientist called Fisher who basically first proposed the structure of these parent porphyrin called porphyrin. According to his presentation, if you see here, this is how basically our porphyrin will look like with 4 subunits of pyrrole. Carbon atom number 1 to 8 will have different substituents attached to them, and these substituents can be either acetate, propionate, methyl and vinyl. There are 2 types of porphyrins, porphyrin 1 and porphyrin 3. So according to Fisher, the carbon atoms should be attached to different substituents, and there are 2 kinds, porphyrin 1 and porphyrin 3. See here, the substituents are attached symmetrically, that is acetate, propionate, acetate, propionate, acetate, propionate, acetate, propionate. So in a symmetrical manner they are attached. If this is the case, then it is defined as porphyrin 1. So in porphyrin 1, from carbon atom 1 to carbon atom 8, okay, the carbon atoms 1 to carbon atom 8, the substitutes that we have are attached in a symmetrical manner. In porphyrin 3, the manner in which substitutes are added is similar, just like porphyrin 1, except for in the ring 4. So if you see here, AP, 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 but in ring 4, it is no longer symmetric. It's PA, asymmetric. It's becoming asymmetric. So this is a major difference between porphyrin 1 and porphyrin 3. Type 3 porphyrins are the ones that are most commonly found compared to type 1. In human, the important porphyrins are of three kinds: uroporphyrins, coproporphyrins, and protoporphyrins. Okay, keep that in mind. Uroporphyrins are found in the urine. Copro, which refers to fecal material. Coproporphyrins are found in feces. Protoporphyrins are found in blood and tissues. See here, Hem, as we have seen, Hem group of the hemoglobin is the most abundant porphyrin, and it will also have 4 pyrrole rings. You can see here with the center iron located at the center. Okay, now you can tell me, see here in a symmetrical manner, MV, MV, MP, but here it's becoming asymmetric in number 4 ring. So what do you think? It's a porphyrin 1 or porphyrin 3? So definitely if it's asymmetric, it's your porphyrin 3, our Hem. Okay, now synthesis of porphyrins begins by the help of two things, glycine and succinyl coenzyme. When the succinyl coenzyme gets condensed with glycine, okay, they will mainly synthesize your porphyrin. And for this, we know that succinyl coenzyme is an intermediate of the TCA cycle or cyclic acid cycle. And this reaction of condensation will be done by the enzyme called ALase synthetase or delta amino levulinic acid. Now, as you can see in this diagram, succinyl coenzyme will get condensed into alpha amino beta-keto adipic acid. And the enzymes that will help will be ALase synthesized, and will also need the presence of glycine. Now, once we got alpha amino beta-keto adipic acid, now this structure will go through a process of decarboxylated to form delta amino levulinic acid. These reactions also will be catalyzed by ALase synthetase, and for this reaction to be carried out, okay, a coenzyme is required, pyridoxal phosphate, PLP. You have learned before from previous lectures what is PLP basically. Now you see here alpha amino beta-keto adipic acid going through a process of decarboxylation, that is removal of carbon dioxide, catalyzed by the enzyme ALase synthetase and coenzyme PLP, and giving us what? Delta amino levulinic acid. Now, 2 ALA molecules are condensed to form the first pyrrole compound known as porphobilinogen. So that delta amino levulinic acid will now become condensed, 2 of them, to form the first pyrrole compound called porphobilinogen. And this again is catalyzed by the enzyme called ALA dehydrase. See here, alpha amino levulinic acids getting dehydrated, removal of water by the help of ALA dehydrase to form the first one, porphobilinogen. Different porphyrins can be then formed from this parent porphyrin called porphobilinogen. So the first porphyrin that developed is your porphobilinogen. The exact mechanism by which this synthesis takes place are not really fully understood. 4 porphobilinogen molecules will react with hydroxymethylbilane, and this reaction will be catalyzed by the enzyme called uroporphyrinogen 1 synthetase. And as it happens, when the porphobilinogen reacts with hydroxymethylbilane, then it can continuously or spontaneously form uroporphyrinogen 1. This is catalyzed by the enzyme called porphyrinogen 3. So basically the whole enzymatic reaction includes two enzymes, uroporphyrinogen 1 synthetase and uroporphyrinogen 3 co-synthetase. Uroporphyrinogens can be then converted into coproporphyrinogens. Later on, coproporphyrinogens can then be converted into protoporphyrinogens. So if you see the diagram, you'll have a better understanding. So 4-hydroxymethylbilane can spontaneously convert and become uroporphyrinogen by removing its ammonia. And in the presence of light, uroporphyrinogen can then become uroporphyrin, or with the help of the enzyme uroporphyrinogen decarboxylase, uroporphyrinogen can convert into coproporphyrinogen 1 or coproporphyrin 1. So the whole reaction also you can see over here, hydroxymethylbilane, the enzyme uroporphyrinogen 1 synthetase and uroporphyrinogen 3 co-synthetase allows it to get converted into uroporphyrinogen 3, which then later can become uroporphyrin. Then the uroporphyrinogen 3 can also become coproporphyrin 3 with the help of uroporphyrinogen decarboxylase. The coproporphyrin 3 can get converts into coproporphyrin 3. The coproporphyrin 3 can also convert into protoporphyrin 3 with the help of the enzyme called coproporphyrin oxidase. The protoporphyrin 3 can again then convert into protoporphyrin 3. Protoporphyrin 3 is the most abundant porphyrin. Among the uroporphyrin, coproporphyrin, this is the most abundant one. It is also the most important porphyrin. Because why? Because heme is synthesized from protoporphyrin 3. Heme group of hemoglobin basically. Heme synthesis is catalyzed by an enzyme called ferrochelatase or heme synthetase. So porphyrin 3 in presence of iron, ferrous iron can ultimately be converted into heme with the help of this enzyme. The heme can then combine with different polypeptides to form different kinds of hemoproteins. Now, this process of regulation, okay, now this porphyrin synthesis is how it is regulated, we need to know. And this regulation is important in order to form heme. So the main enzyme involved in the regulation is our ALase synthetase that develops, if you remember, alpha-aminic levulinic acid. The regulator is the heme itself. Regulation will occur by the process of repression and depression. Let's see how. So when heme is not being utilized, that is when we have sufficient amount of heme in the body. So if it's not being utilized, the concentration will increase. Now as the concentration increases, it will combine with something known as a aporepressor and form something called repressor. These repressor will act on the enzyme that is involved in the regulation called ALase synthetase, and thereby will prevent the synthesis of ALase synthetase. This, if we don't have sufficient amount of ALase synthetase, this will lead to decrease of synthesis of porphyrins. Similarly, when heme is utilized in the body, when the concentration is decreased, the synthesis of ALase synthetase is increased. This will further increase the synthesis of porphyrin. So that's how basically the porphyrin synthesis is regulated. Okay, so we'll stop here today and we'll further continue in the next class with our blood biochemistry here. So for this time, you first must know this whole process. Okay, you should be knowing what is a pyrrole ring, how a porphyrin is, the structure of porphyrin is made up of, how the synthesis of porphyrin takes place, types of porphyrin such as uroporphyrin, coproporphyrin, protoporphyrin, and how this porphyrin synthesis is regulated. It's very important content. Please do remember how the porphyrin is synthesized, the whole mechanism. You must remember, including the enzymes that is involved. So that's all for today. Thank you.